colleagues, I would like to say hello to you here in Vienna, in Austria, for our seminar about new product, our invention. It's a new formulation of a photosensitizer called Phagovir. And I would like to talk today about this product as a COVID-19 prevention mean. So, my name is Andrei Reshetnickov. I'm a Ph.D. in Bioorganic Chemistry and Chemistry of Biologically Active and Natural Substances. So, it's the so-called medicinal chemistry. And I'm here just due to an invitation of the Academy of Holistic Medicine in Berlin. So you can see its contacts. So you are welcoming to watch our materials on the YouTube. So the Academy has its own YouTube channel. And you could also get additional information about other treatments offered through this academy and its activities. So, Phagovir is based on a molecule called pheofetin. So, pheofetin is a derivative of chlorophyll A. After isolation from any plant, like, for example, nettle, so it's Urtica, or spirulina, cyanobacteria, and magnesium atom removal. So in a purified condition, it gets to be formulated with lipids. Those can be natural lipids, mainly phosphatidylcholine, phosphatidyl inositol, just phosphatidylethanolamine present at natural lipid mixtures, for example, derived from spirulina, cyanobacteria, or those can be synthetic or artificial lipids, for example, cholesterol and phosphatidylcholine, ethanolamine, just as mixtures. So both approaches to get lipids from the natural sources or artificial or synthetically, they are possible for this product. The main idea is just to get a finished formulation of pheofetin as micelles or liposomes, so that pheofetin is inside the liposomes and they all form an emulsion. So this emulsion, it's the so-called oil in water emulsion, consisting of multi-layer liposomes. Have a look at pheofetin absorption spectrum. Here you can see 4 main absorption bands. They are marked in green color. The main band is 415 nanometers. Is in blue light. So blue light will activate the pheofetin molecule the best possible way. And the second important absorption band is at 668 nanometers. Have a look, it's the rightmost band on your right hand. It's the so-called red absorption band. What is the practical reason of those two bands? So the blue band is all kind of bright white light sources. It's the daylight. It's the bright LED light. For example, that light is used in the lights of the mobile phones, smartphones. So in fact, that's very convenient to activate phosphatine, and we will learn more about it in the future. And the red light, 668, is a typical light of red LEDs, light emitting diodes. They are often used in physiotherapy. So in many physiotherapy clinics, red light sources like red lights are being used just to warm up, improve the blood circulation. So it makes using phosphatine very easy by both individuals, as all of us have mobile phones, smartphones, same as by clinicians, because many of them have red light sources for physiotherapy. Let's say a few words about the PDT mechanism. Here you can see at the ground with S letter, the so-called singlet or basic level of a photosynthesizer, and on your right hand, the basic triplet level of oxygen. So a photosynthesizer, so for phosphatine in our case, exists in the nature in its singlet state. The oxygen exists in the nature in its triplet state. So the first step is when a photosynthesizer absorbs a photon of light. Then its molecule turns into a singlet, into a triplet. So S1 is aroused singlet state of the photosynthesizer, from which it can just lose its energy and become basic again, or turn into its triplet state of the molecule, so an activated state of the molecule. It's T1 on this drawing. You can find it in the middle, T1. It's the activated molecule of the photosynthesizer after absorbing a photon, a quantum of energy. If this very active triplet state that exists minus 10 seconds, so it's a fraction of nanosecond, yeah, it's a picosecond. It's a very reactive state of the molecule that can quickly react with anything, but the problem is that there are not so many triplets in our organism. In fact, there is only one triplet. Carbon is singlet, nitrogen is singlet, hydrogen is singlet, so there's only one triplet, as I told you, it's oxygen. So there is no other way for this active triplet photosynthesizer to relax its energy, yeah? Only to interact with oxygen. That's a fantastic feature of the nature, and this makes photodynamic therapy possible. This only feature makes photodynamic therapy possible. If it were not for this feature, there would have no been photodynamic therapy at all. So the activated condition or state of photosynthesizer triplet T1 relaxes its energy on the basic oxygen triplet oxygen O2. So molecular oxygen quenches the activated state of the photosynthesizer. And you get an active state of a photosynthesizer called singlet oxygen. So again, as I told you, most of the molecules in our organism are singlet. But now, among those molecules, singlet atom molecules, you got an activated state of oxygen, singlet oxygen. And this activated state of oxygen is higher by energy than other singlet states of other atoms. So what you get? If you get singlet to singlet, it's an allowed transit of energy. Then the energy will go from singlet to another singlet, from higher energy to a lower energy. And there is an energy transmission from singlet oxygen to carbon. And of course, carbon will get plenty of energy, and the carbon-carbon bond will get broken. So It's called oxidation. So we will oxidize and break carbon-to-carbon bonds, and we will also oxidize double bonds, carbon-to-carbon. So as well as double bonds, carbon-oxygen. So all aldehydes, all ketones, all other unsaturated bonds will get oxidized, as well as many bonds, especially in proteins, will get cleaved. The main effect of singlet oxygen reactions is oxidation of proteins. So most of the proteins in the vicinity, in the surrounding of the singlet oxygen reaction will get oxidized. Of course, that will result in the cell lysis and membrane damage. Cell membrane perforation is the main result of a singlet oxygen reaction in the tissue. So most of the tumor necrosis that happen due to a cell lysis, just perforation of plasmatic membranes of the cells. So the cells just leak and generally it looks like a hemorrhagic necrosis of the tissue. Some reactions which may also follow are connected with apoptosis, a delayed cell death that happens in a couple of weeks after photodynamic therapy. So tissue necrosis or immune modulation are main two outcomes of photodynamic reaction of the singlet oxygen and the tissues. We will more talk about immune modulation today because for Phagovir and for the coronavirus prevention, immune modulations plays a more important role than the direct killing of the coronavirus. Yes, the direct killing of the coronavirus is possible too, as you will learn. But the immune modulation is, in fact, a kind of a vaccination. So the immune modulation, in my opinion, is even more important. Summing up, singlet oxygen is not a radical, it's not a peroxide. Singlet oxygen is a molecule of oxygen, it's a molecular oxygen, but it is a higher energy state of oxygen. It is different from other active forms of oxygen, like oxygen radical, like peroxide, like hydroxy radical, or like hydrogen peroxide. Why? Because to get singlet oxygen, you need oxygen just to absorb energy without chemical change or chemical modification of the molecule of oxygen. So oxygen stays oxygen. It's just a higher energy level of oxygen. That's it. What light sources can be used to activate oxygen, let's say, yeah, because we are activating oxygen, in fact. What light sources can be used to activate oxygen? There are mainly three available light sources, as I told you. On the right-hand side, it's right bottom of the slide, you see the emission spectrum of LEDs, light emission diodes. Those are white LEDs, which are used in the smartphones, just as lights of the smartphones. You can see it very well that the emission spectrum of the LED lights, which is a solid line of two colors, blue and violet, it in principle covers the absorption peaks of pheophytin molecule, shown in yellow. So look, most of the four absorption bands of pheophytin are underneath the emission spectrum of the LED lights. So just follow this line with two curves, with the curve with two peaks, follow it, yeah? And the yellow bands of pheophytin are all covered with this, they're all beneath. It means that the LED light from the smartphone will perfectly activate pheophytin. Look at the left spectrum of UVA. It's usual UVA tube for tanning. It's for sun beds, solar panels, tanning cabins, or just the UVA light of the sun of the day, daylight spectrum. So one can just easily use those UVA light sources to activate pheofitine and so Phagovir. So on the top, it's just a mercury luminescent tube. It's just a normal luminescent tube, often used for artificial illumination of just accommodations of rooms inside buildings. Again, you can see that at least two, number 3 and number 4, peaks of its emission spectrum can arouse the molecule of pheofitine. So it's the upper spectrum. So look at number 3 and number 4 yellow peaks. Yeah, number 3 and number 4 yellow peaks. They coincide with the emission bands of the mercury luminescent tubes. In fact, artificial light inside the rooms, sunlight, and the light from the smartphones can all very efficiently activate Phagovir. So they can all very efficiently activate pheofitine, so that pheofitine activates oxygen, and we get a plenty of active oxygen inside our body. Just in case we have applied the product, for example, we sprayed the product in the mouth, it's enough then just to walk a little bit outside while just breathing or talking, or if you're inside, you will get this light from the artificial light sources. If you like to make it stronger, then you can use the lights of your smartphone. So you can shine for 40 seconds in your mouth or for 20 seconds in each of your nostrils, so that you will get pheofitine activated in case you just have sprayed it before illumination. That's the main idea behind photodynamic therapy and behind using this product. So what was the scientific background? The scientific background, in fact, is the method. It's called photoimmunotherapy. It's a well-known treatment modality. In Russia, it was developed by myself and Dr. Vasilyev, Nikolai Vasilyev from Novosibirsk. We applied for a patent in 2006 called photoimmunotherapy treatment in a man by a photosensitizer activated with electromagnetic energy outside the body. So what you need a photosensitizer, like for example this spray, you need light, and you using light, photosensitizer, and oxygen, which is about just around us, we get oxidized molecule of pheofitine itself. You will see how it originates, where it originates from, and this activated molecule can further exist in our body, persist in our body for hours, thus causing oxidation of areas where it accumulates. So this is the patent, Russian patent of 2006. And initially we were mainly aiming at treating cancer and tuberculosis. So we started with cancer patients, but all of you are well aware of the fact that cancer is tightly connected to infection. In fact, infection is the always preceding cancer, and infection as a latent, so hidden, persisting in the body for years, such kind of infection as, for example, Candida albicans, some other fungi, such as mycoplasma, such as toxoplasma, some kind of bacteria. So persisting in tissues for many years, they may cause mutagenic transformations in the tissue, leading to cancer. So initial idea of using this method, this oxidizing action of the photosensitizer photodynamic therapy was initially planned to get kind of damage or transformation of cancer cells. But we soon found that tumors and cancer lesions are full of all kinds of infection. And we could see that those pathogens, they also got oxidized or killed through the photodynamic therapy effect. We will later on during this presentation see it. And through the exposing the epitopes of the destroyed cancer cells and microbes, microorganisms, through the fragments of their shells, they would enhance the T-cell response. Later on, a group of our colleagues from the Netherlands, led by quite well-known, famous immunologist Ferry Ossendorp, you can now see this reference and this paper published in the Journal of Immunotherapy of Cancer in 2018. They used photodynamic therapy using our product to show that if you destroy cancer cell through our product, light, through photodynamic therapy, then the fragments of a cancer cell are able to induce and enhance specific T-cell immune response and tumor eradication, not only in the site of photodynamic therapy, but everywhere, all over the body. So metastasis of tumors, distant metastasis of tumors away from the site of photodynamic therapy also were involved in the breakdown process. So they, of course, demonstrated it in mice, in murine experiments and animals, but they are still now working on transforming this approach into a clinical method. So I think that comes in the future. What was noted during those experiments, that bacteria, fungi, viruses, but not only, but also circulating immune complexes responsible for many autoimmune disorders, histamine and type 2 prostaglandine mediated inflammation as well were all involved in this in those reactions. So that the type 2 prostaglandine mediated inflammation replaced the type 1 histamine mediated inflammation. So they competed with each other, and histamine type mediated inflammation stopped beforehand, being replaced by the second type, type 2 prostaglandine mediated inflammation. So what is the practical sense, practical use? We can quickly stop inflammation in the body caused by autoimmune reasons, like, for example, COVID-19. It's an atypical pneumonia, it's a typical inflammation in the lungs. So by using photodynamic therapy, we can quickly cope with that. It's a kind of solution to that problem. In addition to other important applications like killing bacteria, so eradication of bacteria, fungi, viruses, stopping autoimmune problems like psoriasis, for example, like many eczemas. So what is the scientific background? Peroxide reactions is the chemical background under those transformations. So many studies have shown that the activated oxygen, so singlet oxygen, oxidizes many organic substances, including photosensitizers themselves. So pheophytin that activates oxygen can be oxidized by the same oxygen, but not completely destroyed, but just become a molecule transporting the active oxygen species to the tissues. The pheophytin by itself can accumulate in tumors and cancer cells and in inflamed tissues. It's by itself, by its natural property. So if you deliver pheophytin in the body, through the mouth, intravenously, as a topical formulation like Phagovir, it will migrate in the body by its own pharmacologic properties. It will accumulate and stick to tumors and other pathogenic areas in the body. And together, it will carry on itself an activated oxygen species. So the activated oxygen will travel in the body, in the tissue, together with pH15 molecule. And then, number two, this complex of pH15 and activated oxygen in the tissue will interact with other molecules in the tissue, like proteins, nucleic acids, lipids, and etc. So oxidizing them. Look at next picture, long-lasting peroxides. On the left-hand side, you can see chlorine molecule. So it's a metabolite of pH15 in the body. So if you swallow pH15 or if you just spray it in your mouth, it will convert to the molecule that is on the left-hand side, the first molecule. It's called chlorine E6. So chlorine E6 is the usual metabolite of pH15. And same as pH15, chlorine E6 can get oxidized. You can see it's in the middle, the structure which is in the middle. With oxygen, you can see the pyruvate, so acidic acid moiety in the lower part of the molecule, in the middle of the lower part of the molecule. Maybe we could show it. The molecule in the middle, the structure in the middle. Yes, the bottom, the lower part of the molecule in the middle. There is this OOH. This is OH is just oxygen that attached to the acetic acid residue of this molecule. Why? Because the proton H that is in this methylene position is extremely acidic, and it is easily replaced by oxygen. So you get an organic peroxide that is very stable because the p electrons of oxygen get reallocated on the macrocycle ring of the porphyrin core through the so-called mesomeric effect. So it's just a reallocation of the p electrons of oxygen on the core of chlorophyll molecule, porphyrin ring, stabilizing the whole molecule. So it's an organic peroxide that is extremely long-lasting and long-living. It can exist in the body for hours. So once you get this peroxide in your mouth through just spraying Phagovir and shining using the smartphone light, it will absorb and it will stay in your body, in your tissues for hours, as it is. And then with temperature, our body temperature, it will release carbon dioxide and water and will convert into an oxidated form that is 10 times more effective than its precursor, pH15. So, in fact, metabolites are 10 times more active than the initial compound. So, the superoxide anions get stabilized through p electrons of the porphyrin ring through the 18 p electron system, thus yielding in peroxide stable for at least many hours, sometimes a few days. So the clue of our invention is that intramolecular transition of a reducing electron becomes possible. So reducing electron gets stabilized through an intramolecular transition inside the molecule of chlorine. The same way, the second, so you can see this in the picture, that the same way the second oxygen molecule can attach to another part of this structure. So you can see it on the right hand, just to the right structure, above the upper vinyl group of chlorine. The vinyl group also exists in chlorophyll. So oxidizing this vinyl group through the same attaching of oxygen gives you another peroxide in the same molecule. So now you've got already two peroxides in one molecule. when you get two peroxides in one molecule, so many biochemists know that will immediately result in a dismutation reaction. So dismutation reaction will happen in the tissue, in pathology area, with the release of the singlet oxygen and hydrogen peroxide. Now you get a distant effect from the molecule which you activated in the mouse. So you spray it in the mouse, you shine in the mouse with light, and then in a few hours, somewhere in your body, this dismutation reaction happens with the release of singlet oxygen and hydrogen peroxide all of a sudden. So the viral particles or cancer cells, they never expected this, because just from some unbelievably non-toxic chlorophyll molecule and oxygen and red light or white light or blue light, which are by themselves, they're absolutely safe, non-toxic. But through accumulating this oxidizing potential, they become toxic, but exactly in the area of accumulation. But accumulation of those molecules, as I told you, goes mostly to tumors, inflamed areas, and infected areas. So they don't accumulate in healthy tissues. That's a very important feature that was shown in pharmacological, pharmacokinetical studies in both animals and humans.
Do you know why?
Yeah, there are 5 reasons why. Because of acidic potential of those areas, because the pH is low, pH is acidic. So in tumors, in inflammations, it's acidic, its pH value is low. Then the electronic potential of tumors is very high. Then those areas are full of dendritic cells, leukocytes, phagocytes, like macrophage. They all come to those areas, inflamed areas, tumors, just to work, to clear the necrosis, to kill the infection. And macrophage and other leukocytes, they have fatty membranes, so they're very fatty, oily. And our molecules are oily too. So oil sticks to oil. That's why we used, by the way, it's a good question. That's why we used liposomes. Yeah, exactly. Because without liposomes, the effectiveness was 2 times less. We compared using this stuff just as a solution with detergents like Twins, just usual detergents used in pharmaceutics. So with detergents, it was twice worse than with liposomes. So oil sticks to oil. That's one more issue. And there is still an issue of endocytosis. By some reason, bacteria and cancer cells, they took those molecules and liposomes for food. They think food and just eat it up through pinocytosis. So endocytosis, pinocytosis is a typical how the molecules, thiophitine, chlorines, and those liposomes penetrate the cell. And using by luminescent cytology methods, you can see luminescent microscopy methods. You can see accumulation of pigments of chlorophylls or photosynthetics inside lysosomes. So lysosomes are the stomachs of the cells for digestion, full of enzymes. So you can see accumulation of lysosomes. Why? Just because cells just take those molecules for food, for just food molecules. And that's number 4. And number 5 is that many ingredients, components of the core of a cell are negatively charged and ionic. So they draw cationic photosynthetases. And you can see while you're looking at the molecules that there are 4 nitrogen atoms in the molecule. So those 4 nitrogen atoms get easily protonated at the acidic medium of tumor. And from the blood, which is alkaline, it will quickly relocate into acidic areas where they get protonated. So you get ammonia, secondary, tertiary ammonia ions like NH plus or NH just look at the molecule, you will see this NH plus, just because of attaching of N2H plus and H plus, because just of attaching of additional protons from the medium. So they become positively charged, and through this positive charge, they will then draw to negative charges of the, let's say, mitochondria membrane, because it's negatively charged, and to some structures inside the nucleus, inside the core of a cell. So through this, you will see the accumulation of those molecules, not only in lysosome, but also in mitochondria and in nucleus. And when you give light, you will, of course, cause the damage of lysosome, just mitochondria and nuclei, causing irreversible damage to a cancer cell. It's called apoptosis. So that's the mechanism behind the apoptosis. So thank you very much, Annette, for that question, because it specifies how it works inside the cell and why. Active oxygen particles, first, through a dismutation, will lead to hydrogen peroxide and singlet oxygen. That's reaction number one. You can see it, hydrogen peroxide and singlet oxygen in the tissue. Yeah, that's the reaction number one. It's a dismutation reaction, what I told you about. Then second reaction is when the peroxide, hydrogen peroxide at the acidic media of inflammation or tumor. As I told you, the acidic tumors and inflammation areas, they're often acidic, pH is below 7. So proton plus hydrogen peroxide plus superoxide anion, second reaction, will give you hydroxide anion. Hydroxide anion is extremely active, chemical extremely active. So number two. Number three, the hydroxide radical, hydroxyl radical anion, will then react with superoxide anion, again, giving yield to singlet oxygen. In addition, number four, oxygen can react with singlet oxygen, giving yield to ozone and atomic oxygen. So imagine what is going on in the tissue. It's like an, a nuclear burst. Even atomic oxygen O can appear. So ozone, atomic oxygen, singlet oxygen, hydroxy radical, and hydrogen peroxide. So altogether, they all called reactive oxygen species, ROS. And all they appear in the tissue. And this may be in hours after you use the spray and after you use the light from your smartphone. It will happen deep in the tissue. Sometimes even in tumors that are far away from the initial place where you applied it. So that's the essence of our discovery of this invention. What's going on a cell membrane and the receptor, for you being biochemists, is well known. I think you know very well how plasmatic membrane of the cell looks like, and how a receptor looks like. It's just schematically. At the receptor, many cell markers marking relation if it's your own cell or if it's a pathogenic cell. So all those markers, they look quite similar to what is just drawn here. It's just schematically. And if it's chemically oxidized, if it's modified through those reactive oxygen species, of course, you will change the antigen, the antigen exposing features of the cell, so that the immune system would start to recognize it. What are the advantages of this? This oxidation, it takes place using chlorophylls, derivatives like Phagovir, and light and oxygen. All those reactions are selective. They hardly damage normal tissues and normal cells, just because of the reasons which we were talking before. They don't destroy a cell or change a tissue. There is no necrosis. There is no necrosis. There is no dysplasia in the tissue. There is no damage to blood vessels like thrombosis, typical of classical photodynamic therapy. So during classical photodynamic therapy, when you administer a photosensitizer, it would accumulate in a tumor. Then you give light to this area, the presence of oxygen, then it causes a necrosis. So here there is no necrosis. There is no damage to the tissue. Here, on the plasmatic membranes of the bacteria, fungal cells, cancer cells, a photochemical modification of molecules takes place, which are shown on the right-hand side on the bottom. Those receptors get changed. That leads kind of a change in a recognition and transition of signals through various bioregulators. So the pathogenic cells become recognizable by the immune system, that will better recognize them and better suppress the pathogenic process. Now let's come back to the coronavirus. So with the coronavirus, there are few ways that have been offered to the population for its prevention through the pandemic, just to bring down this dangerous epidemiologic situation which is happening, is going on now in many countries. What is the, let's say, grounds? What are the grounds of using masks, for example? The grounds for using masks have been published in 2021 in Journal of Aerosol Science and Technology. You can see it. So they talk about the masks, that masks are being simple in use, easily available, cheap. That's only advantages. There are no more advantages of masks and wearing masks. What are the disadvantages of using masks? Actually, they are very bad for the ecology because it's a huge problem of actually somehow process used masks. And they report that now the most abundant polymeric pollution of the ocean is not to the PET plastic bags, but because of the masks, which have been already found in oceans in the form of islands. So it's just icebergs of masks already just floating in the oceans instead of plastic bags that were found in the past. So ecology. Then one needs to replace masks every 2 hours. So wearing textile masks forever, like many people like, or putting them in a pocket and then using whenever necessary, or using them for hours is absolutely useless because they will get contaminated and they will then be dangerous while inhaling, in fact, bringing more infection to the body than without the masks. Masks cause a serious harm to the respiratory and heart system of the body because they have an increased respiratory resistance. When you inhale, exhale through the mask, you have to push. And this gives you a load to your lungs all the time, constant load on your lungs while inhaling and exhaling. Masks have low effectiveness just because the pore diameter of the mask is much more than the size of the most viral particles. For example, SARS-CoV-2 virus can easily penetrate through the masks while inhaling and exhaling without any problem because it's much less than the pore diameter of a mask. Only devices having carbon filters in fact, can absorb viral particles. But again, people who prefer those devices, they will face problems with resistance of inhaling and exhale, and they will soon get problems with the heart, lung, respiratory system. In addition, carbon dioxide that is under the mask that we must get rid of, it will again be inhaled, and through inhaling carbon dioxide, we increase its contents in hemoglobin. So when our hemoglobin can't get rid of carbon dioxide, so we get kind of oxygen starvation and deficiency in oxygen. And with the deficiency in oxygen, in fact, we get a brain problem, memory problem, and all other kinds of problems, just because we get poisoned with the carbon dioxide. Gels. Gels were a second solution to block the virus on our mucosa for those who refuse masks. So if you refuse a mask, you can spray or sprinkle gel in your mouth and your nostrils so that they make a film on your mucosa, and this film serves a natural barrier to the viral particles. So they will just drown in the bed of the gel on your mucosa and will not multiply and will not penetrate the mucosa. And there is again a paper published about using gels, nasal powder, Tefix, as an effective barrier against SARS-CoV-2, 2021, by Medical Journal of Science and Technology results. The drawback of using the gels, like all other hygienic products for the mouth, so we can of course use gels, you can use powders, you can use aerosols. So mouth washes, mouth rinses, everything just to rinse and wash your mouth. Yeah. Drawbacks are obvious. You must use all those products every few hours, every now and then. If you use it just once a day, the effectiveness is much less. The effect they bring is easily destroyed by food, by water in the mouth, by enzymes in the mouth. And besides those products, they reduce the sensorics of men, because they mainly suppress the virus through the acidity, because they also, such products, they are acidic and they suppress the virus through their acidity, without any influence on the phagocytosis of our system, immune system. So without boosting our phagocytes, our immune system, and without providing for a long-lasting immune response against the viruses. So generally, it's just a passive protection without forming an active specific immunity. It's just a protection. So number 3, vaccines. Again, there is a fundamental publication giving grounds for using vaccines among the population. It was published in 2021 in Switzerland, Basel Journal Vaccines. So it's a summation of all the experience using neutralizing antibodies of all types of vaccines. So there are all types of vaccines mentioned in this article. All the variants, all the virotypes of the coronavirus are mentioned. So it's a fundamental paper. So the main idea behind using vaccines was to develop the so-called collective immunity against SARS-CoV-2. But the collective immunity is possible, as they said, in case of 60% of population get vaccinated. But then when they couldn't reach the collective immunity at the 60% level, they were about to talk about the 80% collective immunity. Now we are just going to that very goal, 80%. What are the general drawbacks of vaccination? First of all, the fast mutations of the new coronavirus, they are faster than modification of the vaccines. So, as you all know, vaccines were initially developed from the Wuhan alpha coronavirus type. We're now already facing Delta type, as well as Gamma and so on. So, and there is a discussion going on about how those vaccine antibodies neutralize new types of the coronavirus infection. So you can see in the first paragraph of this page, you can see a reference just evaluating discussing the situation, and it's mainly the publication is mainly negative towards general possibility of vaccine of neutralizing the emerging variants of SARS-CoV-2. So they doubt that it's possible using vaccines in this paper. So if you are interested in this issue, you could just read the original paper following this reference. It's a very interesting review. Secondly, for all the vaccines which are being used now, the so-called antibody-dependent enhancement effect, ADE effect, has been proven. It was earlier described for preceding virus, let's say precursor of SARS-CoV-2 for SARS-CoV-1. So again, in the second paragraph, there is another paper, investigation of antibody-dependent enhancement, ADE, of SARS coronavirus infection and its role in pathogenesis of SARS. Look, the paper was published in 2011. So before this current situation with the pandemic, it was already well known that SARS-CoV-1, the precursor of SARS-CoV-2, already, in case any vaccines for those kinds of coronavirus were used in the clinical practice, already were able to cause the antibody-dependent enhancement. And the existing vaccines, all of them, they have not been clinically studied in details on the subject of that phenomenon. So that phenomenon has been well described for SARS-CoV-1, but in case of SARS-CoV-2, it is highly possible with nearly 100% probability, and it's already been proven in many articles. But by some reason, the producers of the vaccines, they never include in the phase 2, even phase 1 protocol, any mentioning of antibody-dependent enhancement and studying that before approving of the vaccines. Thirdly, eventually, even those people were vaccinated who by no means were not to be vaccinated. For example, just by medical indications. For example, many people who were afraid of losing their jobs or who just wanted to stick to their usual lifestyle, all those people, they had to get a vaccine because of the social pressure or because of the, let's say, influence from medical physicians, let's say, from the MDs. So there are many groups of people who must not be vaccinated just by no means. For example, vaccines based on the mRNA, such as, for example, Moderna, BioNTech, the dendritic cells in tissues pick up the mRNA from the vaccine, and they synthesize on its basis the so-called spike protein of the virus. And the viral proteins get to the plasmatic membrane of the dendritic cells, like macrophage, for example, and they form the MHC2 complex for the presentation to T killers, T3 lymphocytes. In fact, it's a standard mechanism of immune response. Other cells, so where the vaccine is injected, it's injected in the muscles. Where will mRNA go immediately? It will immediately be absorbed by the muscle cells, and then it will be immediately used by the ribosomes for the processing and for the production of the spike protein. So where then the spike protein go? It will go to be secreted through the mechanism of the secretion outside the cell. In the matrix, intracellular matrix, macrophage, other dendritic cells will just absorb it. That's essential, that's normal, that's how it goes. Then macrophage, they will cut the spike protein into pieces by enzymatic mechanisms, and they will expose those fragments onto those membranes, onto the membranes. And then MHC complex will be formed just for presenting those epitopes to T3 killers, T cells, lymphocytes. Other cells can present the same proteins as well, but another complex will be formed, MHC1 complex. But in both cases, they will be recognized, macrophage will be recognized, our own dendritic cells will be recognized as alien, as not belonging to our body. Why? Because they are exposing the fragments of alien protein, spike protein. And then vaccine will actually, in fact, orient T3 cells, T killers, towards the normal cells of the body, towards its own macrophage, other dendritic cells. And the first, in the first order, to the cells responsible to the local immunity function. So in fact, vaccination will suppress the local immunity. But what is the local immunity? For example, if there is any inflammatory process in the body, and for example, there is a kind of extra activation of immunity in all those people, and there are plenty of those people among us with autoimmune disorders, with a high reactive of the immune system. I think there are more than 10% of population with autoimmune, with allergies, with autoimmune disorders, with diseases like asthma, psoriasis, plenty of autoimmune disorders with hyperreactivity of the immune system. What is the result of this process in those people with hyperactivity of the immune system, people having autoimmune problems? It's just can cause complications up to the lethal outcome, up to the death. Look, just in the bottom of this page, there is a system in the United States. It's called VAERS virus. So by July 21, nearly 3,000 people died soon after getting Moderna. And more than 7,000 people died soon after getting the BioNTech. These are just immediate deaths, soon after getting the vaccine. Why? Because of the mechanisms which we've now discussed. Because those people, they already had a high level of immune reactivity. And through informing the immune system about the problem in the site of injection, that there is inflammation, there were plenty of dendritic cells in this area, The immune system was informed just still to increase the response. And it was already at its utmost. It was already at the highest level. And through the increasing the immune response, this response just killed the people. Why not exclude those people from let's say, being vaccinated? No, just because there was no proper clinical trial during phase 2. So they couldn't put them on the exclusion list because there was no study, there was no time. They were all in a haste, hurrying up to quickly get it approved. So how we evaluated the effect from after using Phagovir spray, our own product. Obviously, we first of all evaluated that effect on bacteria, so it is the easiest. So we just smeared just oral cavity, took the samples from the oral cavity before using the Phagovir spray and after using the Phagovir spray, and we measured the flora, saprophyte flora and pathogenic flora in the mouth and nostrils of our volunteers. So you can see kinds of the types of bacteria. Some of them are quite pathogenic, and you can see number of people who took part in the study, the level of the flora before and after. So you can see that the pathogenic flora went considerably down after just one application. So you just spray once, trying with the smartphone once, and immediately after this, in a few minutes, the pathogenic flora goes down. It's just bacterial flora. When I'm not talking about viruses, we initially started with bacterial flora. Even bacterial flora is quickly handled. No, it's in vivo. It's phase 2 clinical trial. And you can see the number of patients, 20 for Enterococcus faecalis, 22 for Streptococcus, 24 for Streptococcus viridans, 12 for Staphylococcus aureus, 9 for Micrococcus, 11 for Candida, and 5 for Neisseria. So they were just volunteers having some complaints for sinusitis, tonsillitis, chronic or acute. So we did this study in an ENT hospital. So otorhinolaryngology hospital, specialized hospital for just normal patients who come with complaints on rhinitis, sinusitis, tonsillitis. Just we offered them the spray and just measure the flora before and after. You can see pathogenic flora quickly goes down by order of 1,000 times, 5,000 times. So it's not just twice. It's by order of from 3,000 to 5,000 times. So we nearly get it eradicated just after one application. So photodynamic therapy, in fact, has a very, very profound and strong antimicrobial action. So antimicrobial PDT is very popular, becomes very popular.
Is that also true for nosocomial infections?
Yes, but that's experimental in vitro.
Sure.
Yeah, it is, but in vitro, not in humans. So what other tests? Antiviral activity was our next test. We used a standard express PCR test, which is commercially available. It is not just an laboratory PCR, it's just an antigens express test. Before and after 3 minutes, following using the spray. In all people who were enrolled in this trial, the second test was negative. In fact, we only enrolled those patients in this study who had the first test to be positive. So the first test was positive. Then we sprayed just once. And in 30 minutes, we measured it with the second test. and it was negative. So again, like with the preceding slide with the microbes, you can see immediately after 30 minutes, you see nearly complete eradication of the coronavirus. That's obvious because viruses are much more sensitive to photodynamic therapy. They're much more susceptible to PDT killing. They're easily to be killed by PDT than bacteria. Bacteria is, let's say, through the evolution, they became more stable to all kinds of damage by singlet oxygen, by hydrogen peroxide, by other peroxides, just because it's evolution. They had to develop the protection on their plasmatic membranes against oxidation. And you, being biochemists, you know what kind. It's just the so-called lipid lakes. It's polyunsaturated acid lipids on our plasmatic membranes, which just absorb radiation, all kinds of reactive oxygen species, and this plays a role of anti-aging. So our cell protects against aging. And until this anti-oxidative potential is there, we still resist aging. When this potential becomes lower to resist oxidation, then we start to actually age. And bacteria are the same. They are similar to our cells. They have developed this mechanism to protect themselves, but not viruses. Viruses will easily die immediately upon you just spray and switch on the light. In a few seconds, there are no viruses at all. Second test was we evaluated the macrophage coefficient activity. We just did it in vitro by taking 5 milliliters of venous blood of patients. Then we added, so it was outside the body, ex vivo, in vitro, ex vivo actually. Then we added the lotion to the venous blood just by pressing once pressing to the dispenser of the bottle. So one dose. And we got a sample just in a thin layer in a well on a plate. And then we exposed that to the daylight. Just no even need in using a smartphone, any kind of light. Just in the daylight, we got approximately 1.5 time increase in macrophage activity. Why? Because our dendritic cells, phagocytes, they like ROS, reactive oxygen species, very much. So at the presence, well known, in the presence of hydrogen peroxide and other peroxides, macrophage get boosted. They get hyperactive. So when you spray and use the light from your smartphone, you quickly activate your macrophages all over your nose, nostrils, and your mouth. And those activated macrophages, they're like gods. They get ready to meet the infection. So before you enter any public place, before you take care after a person who are sick, who has the coronavirus disease, just before, use the spray, shine with the light, and your macrophage will be twice more ready to meet the infection. So it will be more available to neutralize the infection and get rid of it. It's, in my opinion, that proves the, let's say, preventive effect of the spray. Next was immunizing action evaluation. We did it on 3 volunteers, only 3 volunteers from people who had the positive PCR test. We additionally took, in addition to the PCR test, we also took blood, venous blood for IgM and IgG, quantitative determination, before, in the middle, and in the end of the month, 3 times. So why 3 times? You can understand it just because first IgM is an acute response, antibodies would appear, and by the end of the month, IgG. So that's quite amazing that already in the middle of the month, one person developed IgM plus IgG, and one more person developed IgG at the end of the month, and the third person didn't develop any antibodies at all. But all they had a positive PCR test in the beginning. So without symptoms, without infecting anyone, they got, after one spraying, after a few seconds of light, in 30 minutes, a negative PCR test. Initially, at first, it was positive. In 30 minutes, it was negative, and no symptoms, no infecting anyone. In the end of the month, of 3 patients, 2 developed immunoglobulin G, so antibodies, without being vaccinated, without symptoms, without infecting anyone. So it means that, in fact, we are getting a kind of vaccination through a killed virus. So if the virus is killed by photodynamic reaction in the mouse, then the fragments of the virus can immunize us in certain cases. When we got a big load of the virus. So if you use the spray and you don't get the virus, then you don't get antibodies. But if you use the spray and you are active in your life, so you go everywhere, you come across the infection, you meet infected people, you get a big load of the virus, still using the spray, you will get vaccinated and you will get antibodies through this without getting sick, without infecting anyone, without getting the disease, in fact. And you will be vaccinated.
So, short question.
Yeah.
The PCR was from blood or from?
From the secret. The PCR was from the secret here, and the antibodies were from the blood. But only 3 patients, simple test, because we were not aiming at big clinical trials here.
But Andrei, any of the PCR tests at the very beginning was positive.
Positive, yeah.
So, assuming the normal immunological system of the body after positive PCR test, you should develop IgA, IgM, IgG.
Yes,
And
sure.
this has nothing to do with the spray.
However, those people didn't develop any more symptoms. That is different.
Sure. But anyhow, if the normal procedure is working,
Yes, sure.
they should develop
They
IgA
should,
and
they should. But what is important, that one of them didn't develop antibodies at all. Initially, he was PCR positive, but he didn't develop antibodies. This happens.
vaccination also.
Yeah, of course, this happens. But to me, what is the most important after this test? Yeah, some of them develop, some of them not, some of them by the essential reasons. So we are not stating here that it's because of the spray. We're stating here that because of the spray, they didn't develop a severe disease symptoms and they didn't infect anyone, because we tested those people in their families. They all had members of their families. It was our intention to see if they are able to infect or not. So no one was infected in the families with them. They had a positive PCR test, and our obligatory condition was that they don't vaccinate, they don't wear masks, they don't keep the social distance, so they stay normal members of the society. So no limitations. So you are PCR positive, but these are just volunteers from friends and families. It's not a formal clinical trial, because a formal clinical trial with such, let's say, requirements must be properly applied to the medical authorities. So we did it not through the health authorities, we did it just through the volunteers among friends and families. However, that was quite remarkable that they stayed healthy, active, no symptoms, and no infecting anyone. That's the most important.
It was one test, one PCR test, or one. So because sometimes,
yeah, 1 and 1
was.
Yeah, it was only 1 in the beginning and 1 after 30 minutes. Yes, of course, sometimes it can be false. No, it was not 1 and 1. No, it was 1. Yeah, again, it was just a preliminary study, but still, in my opinion, it's quite essential. So we couldn't launch a big study like this by the reasons you understand, because to launch a big study like this, you need a permission from the health authorities and you need a protocol. And so far, it's not a medical product. It's hygienic spray for the mouth so far. And it's in the process of the registration as a medical product. So we will soon launch official clinical trials, and I will, of course, tell you of the results later. So, number 4, this was allowed, in fact, to enroll many patients. So we enrolled 3 groups of 30 people each. So altogether, there were 90 people, 3 groups, 30 people in each. Age from 12 to 65 years. And we used the spray at the peaks of the coronavirus pandemic. So there are 3 waves, 3 peaks since 2019 there have been. So during 2020, 2021, during the peaks of the pandemic, we offered the lotion to those 3 groups of people by 30 volunteers each. So they used the spray when they visited public places, when they contacted sick persons with a proven coronavirus, just at their liking, a few times a day. But obligatory, the spray was to be used at bedtime, together with the light, together with the just illuminating the spray. So why 3 groups? One group was control group. One group was using the spray without light as the passive protection as an alternative to a mask. And one group was just using the spray and the light. So the first group of the control group, approximately 10% of this group got the PCR positive in coronavirus. So 10% is an average number of people getting the coronavirus during the peak of a pandemic. So it correlates to just a general occurrence of the COVID-19, or actually of the getting the infection. So neither in the second group nor in the third group.
Getting the infection or getting positive results?
Getting both the positive results by the symptomatically and the positive PCR test, but we used the express test. Some of them, of course, they did additional tests. We didn't put any additional restriction to this research. Who wanted tests from the blood, they just went. Who wanted a laboratory test, they went. But most of them used an express test, antigen test. Then the second and the third groups, they didn't get the virus. So, I mean, the passive protection was fine. And those who used the light in addition to spraying, 10% of them, so those 3 people plus 1 more, they developed in addition the antibodies. But again, we can't now say what was the reasons for the antibodies, just natural way, or they were just caused by using the spray. In the conclusion, I would like to say just a little more about the pandemic situation. It was of summer, but you can see it here that the number of vaccinated people in percents. So you can see it in the upper line, the first figure, upper line, 66.9. It's the percents of vaccinated people in the United Kingdom. So nearly 67. Then column 3, Iceland, 79. And then look at it's in blue color. And then look at the last column but 1, Israel, 67. That's the number of the vaccinated people among the population. So you can see it a lot, yeah, a lot. Then you can go down and see the comparison between the columns marked in blue and the columns marked in green. So the columns marked in green are the countries with easy politics towards vaccination. So where the vaccination was not obligatory or was not let's say, depriving people of certain rights like visiting cafes, restaurants, and firing from their jobs and so on. So those countries shown in green are Ukraine, Montenegro, Slovenia, and Egypt. So, and you can see the vaccination rate in those countries. For example, Ukraine, 9%, Montenegro, 29%, Slovenia, 45%, Egypt, 4%. So compare countries with a highly vaccinated population and countries with a much less vaccinated rate in the population. Now you can see in line 2, the number of sick people just on the day of the study on 10,000 population. So seriously vaccinated Britain, 70%. There are nearly 200 people who got sick in one day on 10,000 population. Compare the UK with the Ukraine, much less vaccinated, it's 4. So compare nearly 200 with 4. Then Iceland, 43 got sick as compared, for example, to Montenegro, 39, Slovenia, 7. Israel, 33 got sick as compared to neighboring Egypt with 4. 10 times less. Then they say that the vaccination prevents from the severe symptoms. Line 4. Britain, severe symptoms on 10 million population, 133. Ukraine, 42. So 3 times less. Iceland, 632. Compare with Montenegro and Slovenia, 9633. Again, 5-6 times less. With severe disease, hospitalized. That's a hospitalization. Israel, nearly 300 compared to Egypt, 10. Then they say vaccination prevents from the death. Line 5. The United Kingdom, for 10 million population, 149 as compared to Ukraine, 38. Iceland, Slovenia, comparable here, but for example, as compared Israel with Egypt, the situation is reversed. In Israel, 82 deaths for as calculated on 100 million population. Egypt, 8 deaths, so 10 times more. In fact, you can see from this comparison, it was just made on one day at the peak of the pandemic, but you can see that all those statements that the rate of vaccination prevents from the severe disease, from hospitalization, and from death, that all those statements, they are fake. In fact, vaccination doesn't prevent. It's an epidemiology study. It's published by the health authorities of those countries and by the World Health Organization. So it's an official information. You can see just statistically, epidemiologically, the level of vaccination correlates with severe symptoms, hospitalization, and death. The more vaccination, the more hospitalizations, severe symptoms, and death. The less vaccination, the less. But it's not just in 10% or 20%. The difference is in times, 10 times, 5 times, 6 times. So it's an absolutely clearly seen from here. Israel, just let's go quickly to Israel because we are finishing now. It's just detailization for Israel only. So you can see from the 25th till the 31st of July 21, the left column is ages. Then the second column, in 1 week, by ages, people who got sick among vaccinated people. So let's here mention that those are twice vaccinated people with Moderna. Then compared with column 3, non-vaccinated people. And column 4, the ratio of vaccinated people among hospitalized. Compare. And the last column, the total percentage of vaccinated people in this age group. On the bottom line, the last line on the bottom, you can see, so among vaccinated people, more than 7,000 people got the disease. Among non-vaccinated, 1,500. Among people who got to the hospital, 86% were vaccinated. And the rate of the vaccinated population among all the groups from 20 to 90-year-olds was 84. So among the 84% of vaccinated population, 85 nearly, 86 got to the hospitals. So when the rate of population, vaccinated population who gets to the hospital is higher than the rate among the population, it means the so-called negative effectiveness of a vaccine. It means, here you can see, by a few percent, more vaccinated people got to the hospitals than they were totally vaccinated. It means a negative effect of the vaccine. It means that vaccination promotes the severe disease. So it actually causes severe disease. It's just one of the laws of epidemiology, the so-called negative effectiveness of vaccine. So we expect positive effectiveness. We expect that it prevents, but it facilitates getting sick. So it makes us easier to get the infection, the same infection. That's unbelievable, but that's true. In fact, what was our challenge in conclusion? It was just to develop a product that could be effective in people, which could allow for both passive protection through using the lipid film, as well as the active protection using the reactive oxygen species and photodynamic reaction to kill or destroy the viruses on the mucosa, which is lipophilic, easily attaching to the viral particle, easily penetrating cells, easily penetrating healthy mucosa, which can be activated with all possible kind of light sources, daylight, artificial light, lights of mobile phones, which can be preventive towards the virus, and which can be also helping people who were vaccinated, because vaccinated people, as we have just discussed, they are more susceptible to getting the virus. That's a paradox. But those who are vaccinated, they will sooner get the virus, they will sooner get the severe disease and death. So we are now aiming at protecting them, first of all, because those who are vaccinated, they will develop antibody-dependent infection, and they will be more susceptible to take the disease. So we are now offering a way to protect them as well. That will also protect against microbes, against other seasonal viral infections, and also against the so-called heterologous immune responses connected with the cross reactions of T3 lymphocyte, T3 killers. Because if you follow this reference of 1980, you will read about mechanism of the cross responses in the immune system actually leading to antibody-dependent enhancement. And we have now all reasons to assume that photodynamic therapy can actually resist the antibody-dependent enhancement. So that's important for vaccinated people. That's what we think now. If you just read the article and think of other references which I gave you, you will probably come to the same conclusion. So I would like to thank Viktor Tsyganov, President of the Academy of Holistic Medicine in Berlin, for allowing for this my presentation. Also, I would like to thank Herman Otto, the Managing Director of the same Academy, for the assistance toward this report. Thank you very much for your attention.
Thank you very